Welcome back to MLT 120. Today we're going to be talking about inclusions in our erythrocytes, or things that we might see in our red blood cells. We're going to be using our atlas today. We're going to be on pages 108 through pages 113. And also at the end of the lecture, we're going to be talking about different artifacts you might see on your slides in peripheral smears. And we're going to be on pages 217 to 218. The first inclusion that you can see in your red blood cells, or your erythrocytes, that we're going to discuss is called Hal-Jolly bodies. These are these little purple dots inside the red blood cells that the arrows are pointing to on images A and B. These can be anywhere from dark blue to purple, and they're usually round to oval in shape. And they're usually somewhere along the edge, but they're not out here on the periphery. They usually are inside the red blood cell, but they're usually not smack in the center. They're still kind of hanging out on the outer rim of the red blood cell. And these are made up of DNA. So their composition is DNA. And here are some of the diseases or conditions that are associated with red blood cells that get Hal-Jolly bodies inside of them. And all this information is on page 108 in your atlas. Now let's move on to page 109 in your atlas, and let's talk about basophilic stippling. And basophilic stippling looks like a bunch of little dots, and again they can be anywhere from dark blue to purple. And you see some examples of basophilic stippling in both images A and B. They are usually numerous in number as far as how many is in the cell or red blood cell, and they're fairly even in distribution. So basically they make the red blood cell just looks speckled with the little purple or bluish dots. And these are composed of RNA. Where the Hal-Jolly bodies was DNA, the basophilic stippling is RNA. And some of the conditions that are associated with basophilic stippling are listed here. One of the main causes is lead poisoning, so make sure you remember that. That's one that they love to ask on the certification exam, is what's a condition that is associated with basophilic stippling. So make sure you file that away and remember that. On page 110 in your atlas, we're going to look at Pappenheimer bodies. These are also known as siderotic granules, so sometimes you'll hear those terms used interchangeably. And those are little bundles or clumps in your red blood cells, and you see some examples here on figures A and B. And on figure C, they're referring to it as the siderotic granules. But these are usually light blue in color, and the composition of your Pappenheimer bodies are iron. So these guys are made up of iron. And here are some of the diseases or conditions associated with the basophilic stippling, but just remember these usually come in clusters, granules in clusters. Next up on page 111 is our Cabot rings. Cabot rings are thought to be remnants of the mitotic spindle. Remember, the mitotic spindle helps our cells divide, and these are kind of a rare finding. And the Cabot rings are these kind of fine, spindly little rings that you find. Sometimes you can find them in figure 8 patterns, but these are not found very often. They're a rare find, but again, they're thought to be a remnant of the mitotic spindle. And sometimes the Cabot rings are confused with malaria, and that's because the parasite Plasmodium that causes malaria will also cause a ring to form inside your red blood cells. So sometimes that can be confusing to a lab tech when you are looking at a smear and you happen to see a ring inside your red blood cells on a patient. You might think, oh, this is malaria, but it may actually be a Cabot ring or vice versa. So just be careful with that. You might need to look at patient history to see if a patient's been out of the country. It might be malaria, or it may be a Cabot ring. But just remember, this is a kind of a rare find. We will be looking at the structure of that ring that forms for malaria in MLT 240 special micro. So at some point in your time with us, you will be looking at that malaria ring. On page 112, let's look at some inclusions that are in your supravital stains. All the slides that we've looked at previously have been stained with a right stain, and that's the stain that we normally use in hematology, but we can also use a supravital stain. So these have been stained with new methylene blue, which is a supravital stain. So let's look at figure A here. In figure A, we're looking at reticulocytes. So if you remember, reticulocytes is the step right before mature red blood cells. So usually reticulocytes kind of have that bluish purple color to them anyway, but they will appear kind of bluish colored because of the stain. If you're using a supravital stain, in particular the new methylene blue. But also something that you'll be able to see in the supravital stain that you won't be able to see in the right stain with the reticulocytes is the remnants or the precipitant of the RNA. And that's what these kind of specks or stringy looking stuff is inside the reticulocytes, is the RNA. And I think we talked about it before, but you have to use a supravital stain in order to see the remnants of the RNA in the reticulocytes. Okay, so that's what you're looking at in figure A. Okay. Over in figure B, let's hop over here. You're looking at Heinz bodies. Now Heinz bodies look very similar to Hal Jolly bodies, but Hal Jolly bodies are inside the red blood cells, where Heinz bodies are kind of hanging off the edge. So make sure you make that distinction. See, all these are hanging off the edge, hanging off the very edge of the red blood cell. Now, it'll say that you need to use a supravital stain to see the Heinz bodies, but you should be able to see them on a right stain as well. And the Heinz bodies are made up of denatured hemoglobin. So with the Heinz bodies, this is when the spleen will see that the red blood cell looks abnormal because it has this Heinz body hanging off of it. So when the spleen sees that, guess what it's going to do? It's going to pit this red blood cell and it's going to take a chunk off of it. Okay, and when it takes a chunk off of it, it's going to become a bite cell. Okay, remember when we talked about the red blood cell fragments, and a red blood cell can become a bite cell? That's one of these situations, is the spleen has identified this as abnormal, this Heinz body is abnormal, and it's going to take a chunk out of this red blood cell to get rid of that Heinz body. Okay, down here in figure C, these are showing you hemoglobin H, and again this is stained with a supravital stain, new methylene blue. These are shown to be, or described to be, like a raspberry or a golf ball. Okay, I want to show you this chart on page 113 of your atlas. This is a really good chart to help show the different inclusions. Here on the far left hand side, what the inclusions are made up of. Okay, so what they're composed of, and which stains that they'll show up on. So here is our right stain. Here's our supravital stain, our new methylene blue, and our Prussian blue. And this is our iron stain. And really the only one that it's associated with is that Pappenheimer body, that you're looking for that iron, because the Pappenheimer bodies are made up of iron. Okay, so that's really the only one that's significant for. But remember that, because that's another certification exam question. It'll ask which or what inclusion that Prussian blue is looking for, and it'll be Pappenheimer bodies. It does say here that the right stain, you can't see Heinz bodies, but I've seen right stains where people have seen Heinz bodies. So that's the only thing I kind of disagree with on this. So if you see a 0, it's saying that you won't be able to see it. If you see a plus sign, you should be able to see that inclusion with that particular stain. So this is a handy little chart. Now, if you will, flip over to page 217 in your atlas. We're going to talk about some different artifacts that you can possibly see on your blood smear slide that you guys make to observe under the microscope. So let's look at figure A first. Figure A is looking at precipitant from your stain. So we use different stains, of course, to stain our blood smears. Of course, you can see your red blood cells here, but all this like greeny looking purple sediment is precipitant from your stain. So this is things that are coming from within your stain that is sitting on top of your cells. Okay, so this would be considered an artifact. And this could be a result of the stain getting too old or getting close to the expiration date, or just the stain being dirty and needs to be changed out. Next up is figure B. This would be bacteria on your blood smear slide. Of course, if a patient has bacteria in their blood, that's clinically significant, because bacteria should not be in a patient's blood. But as far as making a blood smear slide and for counting cells, it would be considered an artifact for counting cells purposes. And if your red cells are in focus, the bacteria should be in focus. Moving down to the bottom of 217, looking at 22.17, the figure, we're looking at a drying artifact on these red blood cells. These very refractile looking bubbles that are on top of our red blood cells, these are a result of drying. And again, it's highly refractile, meaning that it is shiny when you focus up and down on the blood cells. Now let's look over at figure 22.18, and we're looking at a clump of platelets right here in the center. When you have a clump of platelets, that can be a problem for a patient sample, because a clump of platelets on an automatic hematology analyzer, this clump of platelets can register as other types of cells, making your counts inaccurate. So if you end up having a lot of platelet clumps in a sample, you may end up having to call and get a redraw on a patient, because if you have all these platelet clumps, you don't want your counts to be inaccurate. So that's something to look for, but a platelet clump would be considered an artifact. The last artifact that we're going to look at is on page 218. This is called platelet satellitism. This is when your platelets clump around your neutrophils. Okay, see all these little platelets are kind of surrounding your neutrophil. Remember, our neutrophil is one type of our white blood cell, and all of our platelets have come and gathered around our neutrophil. So that's called platelet satellitism. This concludes our lecture on red blood cell inclusions and artifacts you may see in blood smears, and I will see you over in the next lecture.